Hello and welcome. So today's topic is the microanatomy of the liver. It is a very tough topic to understand in first year MBBS. So try to concentrate. I will try my best to make you understand. Okay? You know that liver is the visceral organ in the abdomen. First of all, suppose this is the liver and the primary layer of the liver or primary covering of the liver is the Gleason's capsule. Then you're gonna find visceral layer and then you're gonna find parietal layer. Okay, now the entire liver is consist of 20% vascular structure and the rest of 80% is parenchyma. Now the vascular structures are basically the hepatic artery, hepatic vein, then central vein along with the portal vein, along with the lymphatic vessels and the nerve structures. Okay, and parenchyma means the parenchyma of the liver is made by type 3 collagen, basically the reticulin connective tissue. Okay, now another point, as I told you in my liver anatomy videos, that liver is also known as hepato or hepatic organ. So the cells inside of the liver is known as hepatic cells, or in shortcut hepatocytes. Now all these hepatocytes remains in the liver as a cluster formation, and this cluster has three groups. First of all, hepatic lobules or classic hepatic lobules, then you're going to find portal lobules, and lastly you're going to find liver acinus. Okay, first of all I'm going to talk about classic hepatic lobules. Okay, as you can see in this photo, so you can see this structure from above. Okay, it is a hexagonal shape, but you can see here it is a 3D shape. Okay, now at the center you can see the central vein. It is also known as centrilobular vein, but you don't have to remember this tough name, so remember this shortcut name, central vein. Okay, look at this, each corner, this is a hexagonal structure, so it has 6 corners. Each 6 corners has portal canals. Each portal canal consists of hepatic artery, bile duct, portal vein and hepatic lymphatics. So we are getting 4 structures. So we can call it portal tetrad. Okay, but we intentionally ignored these hepatic lymphatics. Why? Because this is very thin and tiny structure and it is difficult to see under the microscope. But anyway, so we intentionally ignore these hepatic lymphatics, so we can call it these 3 major structures as a portal triad, as you can see in this photo. Now you are getting one portal canal which consists of portal triad. Now one portal canal supplies the 3 adjacent portal or the hepatic lobules. Okay, again in this hepatic lobule you can see that the cells or the hepatocytes are spreading towards the periphery, towards the boundary of the hexagon as a plate shaped or a sheet like structure. So this phenomena we call it hepatic lamina. Each lamina consists of 20 hepatocytes. Now there are multiple hepatic lamina. Okay, the space or the gap area in between this hepatic lamina is known as hepatic lacunae. Lacunae is a gap area. Okay, so a gap area or a space in between 2 hepatic lamina is known as hepatic lacunae, and this hepatic lacunae consists of hepatic sinusoids. Now this hepatic sinusoid has mixture of blood coming from hepatic artery and portal vein, because the portal vein carries the all nutrition from the intestine and hepatic artery carries the oxygenated blood. So all this mixture presents in the hepatic sinusoid and it delivers through the hepatocytes by a diffusion process. Okay, so what happens, this oxygen and all the nutrition components utilizing by the hepatocytes again enters into the sinusoid via diffusion process. It's a continuous in and out process. So the mixture of this blood in sinusoid ultimately drains into the central vein. Okay, now each central vein goes outward and connects with other central vein of the adjacent lobules. Okay, so this multiple central vein connects together and forms interlobular vein. Okay, now each interlobular vein connects with other interlobular vein and forms ultimately hepatic vein. Now this hepatic vein ultimately drains into inferior vena cava. Okay, sinusoid presents in the hepatic lacunae. Lacunae means a gap area or a space in between 2 hepatic lamina. Okay, now the wall of these sinusoids are actually fenestrated, thin and flat. You can find 3 types of cell large cells, okay, big size cells in this sinusoid. First of all, Kupffer cell, okay. So Kupffer cells are actually belongs to the mononuclear phagocyte system, okay, or MPS. I will talk about these things later in pathology chapter, not here. Secondly, you're gonna find Ito cells. These are stellate shaped, okay, Ito cells. And thirdly, you're gonna find pit cells, okay. These are actually modified forms of natural killer cells, NK cells. Again, I will talk about these cells details in pathology chapter, not here. Look at this photo. You can see there is a space or gap area in between the sinusoid and the wall of the hepatic lamina, okay. So this gap is known as space of DC, okay. And again, concentrate in the part of the portal canal. You can see there is a gap area or space area around the portal canal. Now this gap area is known as space of Moll, okay. Now look at this photo and try to understand that, as I said, one hepatic lamina contains of 20 hepatocytes, and this is actually superior view or view from above. Now if you see this from lateral view, you can see the multiple rows of each 20 numbers of hepatocytes as a row or forming a column, okay. Now here is the thing. Suppose this is one lamina and here contains 20 numbers of hepatocytes. From each hepatocytes, one bile canaliculi is arising, okay. Suppose here is one bile canaliculi and it is connecting with this adjacent hepatocyte. Again, its bile canaliculi is connecting with this. Basic calculation, 20 bile canaliculi is arising from 20 hepatocytes. Each bile canaliculi connects with each other and forms one bile ductule. This bile ductule enters into the bile duct, okay. So all these topics I talked about, these are part of classic hepatic lobule. Gone. Second point, portal lobule, okay. See, the portal lobule is basically, it's a imaginary triangular territory. As you can see in this photo, it's a imaginary triangular territory. It doesn't exist in reality, but for our better understanding, I am talking about this is a imaginary triangular territory. At the center of this triangle, you are getting one portal canal, okay, and the triangles are consist of central vein of adjacent 3 hepatic lobule. This hepatic artery gives its branches to these adjacent 3 hepatic lobules. This portal vein gives its sinusoids or its branches to these adjacent 3 hepatic lobules. All these bile canaliculi or the bile ductules coming from these each hepatic lobules and draining into this bile duct of this portal canal, okay. Remember, this portal lobule is also known as nutritional lobule because in the liver, ultimate nutrition is getting from the intestine via the portal veins, okay. Now our last component, which is liver acinus. It is also known as liver acinus of Rappaport, okay. As you can see in this photo, you are getting 2 hexagonal hepatic lobule. Now I am zooming this photo and you are getting 2 hexagonal boundary or the boundary of these 2 hepatic lobule each side, okay. In between these 2 wall of this hepatic lobule, you can see the branches of hepatic artery, branches of portal vein are present in this area, and the bile ductules are going to the portal canal, okay. Each of these transverse vessels forms a backbone which provides the ultimate nutrition and collects the bile secretion from its diamond shaped area, okay. As you can see in this photo, it is a diamond shaped area. So basically it's a liver parenchyma and we call it liver acinus of Rappaport, okay. It is actually divided into 3 zones. First of all, adjacent or the nearest structure is known as zone 1, then you're gonna find zone 2, and lastly towards the central vein you're getting zone 3, okay. Now common sense, this hepatic artery and the portal vein giving branches to these each wall of these hepatic lobules, and the nearest zone is getting maximum amount of oxygen and nutrition because the hepatic artery and the portal veins are the nearest structure, okay. Now after from this zone and diffusion process by the sinusoids, this oxygen and nutrition will go to the zone 2, which will be lower, okay. So zone 2 are getting moderately low amount of oxygen and nutrition. And then it will go to the zone 3. So the cells or the hepatocytes of this zone 3 are getting least amount of oxygen and nutrition. Okay, so what are the clinical conditions are related with these zones? I will talk about this thing in medicine chapter, because there are lots of disease and ICU conditions and critical conditions are there during the blood supply shock, acute blood loss, then hypovolemic shock, and there are lots of metabolic conditions. These zone 3 and zone 2 cells are getting affected. Anyway, I hope you can understand a little bit about this microanatomy of the liver. So if you understand, then like it, subscribe it, share it. Anyway, see you soon. Till then, bye.